Pharmaceutics, Punjab University, Chandigarh, India. And ma'am will be giving us a talk on novel targeted theranostic docetaxel curcumin co-encapsulated lipidic nanoparticles for breast cancer, in vitro and preclinical proof of concept. Over to you, ma'am.
Thank you. At the outset, I would like to thank the organizers and a very good afternoon to all of you. I am a person who is a formulation scientist, and my predecessor, the previous speaker did mention the issues with breast cancer, the epidemiology. So I am looking at the options, how we can improve the efficacy of these anti-cancer molecules and also avoid the side effects. So with this intent, we started with this study. The issues with docetaxel, we all know it is otherwise a widely prescribed anti-cancer molecule, but PGP flux and resistance are very common. And the marketed product is because most of these anti-cancer molecules have a very poor solubilities. So the options are that certain solvents are being used, and the toxicities associated with these molecules are, one, because of their natural nature, because they also harm the normal cells, but in addition to that, sometimes the way they are formulated and the type of solvents which are used could be toxic. So the solutions I offered was that if I can combine docetaxel, first of all, if I can put it into a carrier system, which would improve its bioavailability and also maybe target it to the specific site. And secondly, if I can add a molecule, which would be a PGP flux inhibitor, and preferably of a natural origin, then maybe the efficacy can be enhanced. And that's what we did. We encapsulated docetaxel along with curcumin taken as a PGP flux inhibitor. We all know curcumin has its own anti-cancer profile as well, but here we used it at a much lower doses, where we were considering it to be a chemosensitizer, where it would improve the efficacy of docetaxel. And we put them into solid lipid nanoparticles. Why the lipidic nanoparticles? One, they are considered to be safe versus the polymeric nanoparticles. Liposomes have been common and on the terrain for quite some time, but there have been limited success stories, and the reasons are they are not very stable, they are leaky, and they are difficult to commercially scale up. So all of these can be taken care of if we are using a SLN system, which is the solid lipid nanoparticle system. They are scalable. The equipment are usually available with the pharmaceutical companies, and the stability, biocompatibility of the lipid are also very common. So with this, we took up, and what were we going to achieve is improving the bioavailability both of docetaxel. Curcumin is also a tough molecule. It is not bioavailable, with the reported bioavailability being less than 1%, and it is also highly prone to degradations and metabolism, fast metabolism. We had previously developed solid lipid nanoparticle technology for curcumin, and patent, Indian patent is granted yesterday only to me, and in addition to that, we've also filed these patents across US, Canada, and Europe, and we are looking for those results as well. So we had established these SLN carrier systems to improve the solubility, stability, and efficacy, also protect it against photodegradation. Then we wanted to avoid the vehicle-related effects, and these solid lipid nanoparticles are totally aqueous in nature, and that's why we developed them. And the prepared SLNs were autoclavable, so we can get sterile formulations which would be required for IV parenterals. So this is how they look at. So this is the type of dispersion we end up getting. This is what we observe under the transmission electron microscope. The sizes were in the range of 150 to 200 or so, and the charge was also near neutral or between 10 to 20. And this is the release profile. We compared it with the marketed formulation of docetaxel, which is Texotier. And the Texotier was found to release within the 12 hours, while we could extend the efficacy when only docetaxel was put into nanoparticles or when we combined docetaxel with curcumin. The red line shows the release profile of curcumin in the nanoparticles. Then we evaluated the formulated products in a range of cell lines, including the triple negative, that is MDA-MB, then T47, the double negative, and finally we also did it in estrogen and progesterone and glucocorticoid receptor sensitive cell line MCF7. When we used free DTX, that is docetaxel, we could not get an IC50, and MIC was observed at 160 nanometers, while when we incorporated this docetaxel into nanoparticles, we achieved MIC of 160 nanometers, nanomoles in case of the MDA-MB cell lines, which reduced to 80 nanomoles for T47, and for MCF7 again it was 160. When I combined it with curcumin in a ratio of 10:1, the IC50 reduced to almost half or 1/4, with it being 80, with I think 8 nanomoles of curcumin, and it was reduced to 20 for both the T47 and MCF cell lines. So from a MIC of 160, we reached to an IC50 of 20 nanomoles when we combined docetaxel with curcumin and we encapsulated it into solid lipid nanoparticles. Further, for targeting, we added a ligand, which is RGD peptide, over these nanoparticles, and the IC50 values were further reduced to 40. While we could not achieve the IC50 in case of T47, where we found probably it doesn't have those receptors for the peptide, and plus the steric hindrance created by coating the nanoparticles with peptide created a problem. And so we did with docetaxel alone with peptide loading, and very encouragingly, when docetaxel plus curcumin nanoparticles were loaded decorated with a peptide, we could reduce the IC50 to 10 in case of MDMB cell lines. So around 2.5 to 400 times better anti-cancer activities were observed in terms of IC50 values. Then we wanted to confirm that the effects we are achieving is actually because the SLNs are acting as true carriers and they are carrying across the drug within the cell. So what we did was we used FITC probed nanoparticles, and here I show that in all the 3 cell lines, when the nanoparticles were there or nanoparticles along with the peptide were there, the extent of uptake into the cells was significantly improved, and we show the data for MCF in terms of the intensity. Then we moved on to do some preclinical studies where we first performed the pharmacokinetic studies. We administered docetaxel along with curcumin into the nanoparticles, and only docetaxel solid lipid nanoparticles, and we compared it with Texotier, and we found very beautiful effects where the AUC values were more than 10 times enhanced, the area under the curve. The values obtained for Texotier were around 2700, which improved to 24,000 when curcumin was also used along with it, and the mean residence time also increased by from 2 hours to almost 20 hours, and the relative bioavailability was around 8 times. So 800% enhancement in the bioavailability was observed. So this data again shows the curves where we show the AUC data. And then we also determined the levels of docetaxel in various tissues, the kidney, liver, and the spleen. As expected, the levels of docetaxel when Texutier was administered were the highest in the liver, and we know of the hepatotoxicity which is observed with docetaxel. But when we administered it as solid lipid nanoparticles, the values were much lesser in the liver at initial times of around 15 minutes and then also at 2 hours. And there were levels at 2 hours in the kidneys where it indicated that the nanoparticles were fed into the kidneys for being excreted, where the smaller particles would be filtered out and the larger particles would recirculate for extended effects. Then we performed the pharmacodynamic studies where we got beautiful results. These are the size of the tumors when the breast tumors were induced in the animals, and when we treated the animals with docetaxel, which was incorporated into solid lipid nanoparticles, the size reduced, which further reduced when curcumin was also included, and when the peptide was included, you can see the size of the tumor almost reduced. And at times when we were extracting the tumors, we felt that it was just a scar which was left behind because of injections, etc. of DMBA. This was a DMBA-induced model, and similar results were observed with the histopathology studies also. And there was a 106 times reduction in the tumor volume from a 1054 millimeter cube to it reduced to 10 in case of peptide decorated nanoparticles which had both docetaxel and curcumin. For alone docetaxel also there was a 10 times reduction, and there was a 5 times reduction from 1054 to 211. It increased to almost 10 times when curcumin was included, and when the peptide was decorated, as I discussed, it improved further. We also did a marker CA15-3, and again in that case we saw similar results. Then we wanted to evaluate these nanoparticles for a theranostic application where we used some quantum dots, which were again novel in the sense that they were coated by certain zinc and cadmium molecules, which were ensured to reduce the toxicity of these quantum dots. And so the concept is that you have a nanoparticle which has both docetaxel and curcumin, and then you decorate it with the quantum dots, and then these quantum dots are connected with the peptide. So these were all chemical changes we did, and we confirmed by various spectral studies that the particles were decorated with the quantum dots, and there was some quenching of the fluorescence of the quantum dots when they were coated onto the nanoparticles, as was expected, but the level of fluorescence was sufficient to be monitored. And we saw a good stability by these nanoparticles. And when we administered these nanoparticles, I don't know if the picture is very clear, when we administered these QD and peptide decorated nanoparticles into the tumor animal, into the animal in which tumor was induced, and we dissected the tumor tissue, we could see fluorescence in the tissue, which indicated that we can use these as theragnostics. A very interesting observation which was observed when we treated the certain cells, I show here the data for the MCF7 cells, that we were trying to capture images of these cells, the DAPI stain for the nucleus, and we had a fluorescent probe and a QD probe, because QD also fluoresces. And 1 hour post treatment, we saw these connections developing between these cells which were treated by these nanoparticles. Similar connections were also observed when we treated these cells with FITC labeled solid lipid nanoparticles. So this is a sort of a hypothesis. Maybe we need to do some more studies. We all know that tunneling nanotubes are a concern, and they are said to be responsible for metastasis. And there are now reports where they say that these tunneling nanotubes also carry the lipidic cargo from one cancer cell to a cancer lying away from the site for the communication as well as for transport of food, and that's how they achieve the metastasis. So the concept is that if these tunneling nanotubes, once they develop, can be used to target, if these solid lipid nanoparticles act as a cargo and they are transported across these TNTs, then we could achieve a very efficient means to manage metastasis and metastatic cancer cell lines, and a smaller dose could be transported across the entire cancer cell domain, and it could help us manage the disease better. So with this, I end my discussion. Thank you.